All right. Hi, everybody. My name is Chase Minardi, and for my cell biology final project, I chose to cover Wilson's disease. So, introduction. Wilson's disease, also called hepatic lenticular degeneration, is a rare genetic disorder that affects how the body regulates and processes copper. Copper is an essential trace element needed for many enzyme functions, including those involved in energy production, antioxidant defense, and neurotransmitter synthesis. However, free copper is highly reactive and can generate reactive oxygen species, which damage cellular components such as membranes, proteins, and DNA. Normally, the body tightly regulates copper balance. Dietary copper is absorbed in the small intestine, used in metabolic processes, incorporated into a transport protein called ceruloplasmin, and excess copper is excreted into the bile. In Wilson's disease, mutation in a gene essential for copper transport disrupt this balance, causing copper to build up in the liver and other tissues over time. This buildup leads to oxidative stress, cellular injury, organ dysfunction, and potentially fatal complications if left untreated, according to Medline Plus, as well as Ferenczi, published in 2023 and 2022, respectively. Wilson's disease is caused by a mutation in the ATP7P gene located on chromosome 13. This gene encodes a P-type copper transporting ATPase, a protein responsible for incorporating copper into the ceruloplasmin and excreting excess copper into bile, published by Kodo in 2020. More than 800 pathogenic variants of ATP7P have been identified, including missense, nonsense, frameshift, and splice site mutations. One of the most common mutations in individuals of European ancestry is H1069Q, which disrupts the protein's function by substituting a single amino acid. This disease follows autosomal recessive inheritance patterns, so individuals must inherit two defective ATP7P alleles to develop the condition. Carriers with one mutated copy usually do not experience symptoms, but can pass the mutations to their children. Rarely, pseudodominant inheritance occurs in populations where the mutation is common, but the classical pattern remains recessive, according to Medline Plus, as well as Ferenczi previously mentioned. Genetic testing is a valuable diagnostic tool, particularly for family screening and identifying pre-symptomatic individuals, as it allows for early intervention before significant organ damage can occur. Molecular mechanism. The ATP7P protein is located mainly in the trans-Golgi network of liver cells and plays a central role in copper regulation. Under normal conditions, ATP7B has two key functions. It incorporates copper into apoceruloplasmin, forming active ceruloplasmin for copper transport in the blood, and it transports excess copper into the bile canalically, where it is excreted from the body, according to Bull, published in 1993. When ATP7B is defective, these processes fail, leading to copper accumulation within hepatocytes. Excess free copper catalyzes the production of ROS through redox reactions. ROS causes oxidative stress, which damages lipids, proteins, and nucleic acids, leading to cell dysfunction and death. Mitochondria are particularly sensitive to copper-induced damage, resulting in reduced ATP production, energy deficits, and activation of apoptotic pathways. As a hepatocyte die, they release stored copper into the bloodstream, where it can deposit into the other organs, including the brain, cornea, and kidneys, according to Roberts and Shishkley, published in 2022. A well-known example of a harmful mutation is C1934T G, which disrupts normal mRNA splicing and produces a pronounced non-functional ATP7B protein, according to Candalis, published in 2020. These molecular changes lead to a perpetuating cycle, impaired ceruloplasmin production, reduces copper transport capacity, increasing free copper, and worsening tissue damage. Cellular pathology. At the cellular level, Copper accumulation first occurs in the nucleus and cytosol of the hepatocytes, where it begins disrupting normal cell function even before visible cell tissue damage is present, according to Husser, published in 2006. Free copper reacts with oxygen to generate ROS, which damage cell membranes, proteins, and DNA. This oxidative stress leads to mitochondrial dysfunction, impairing ATP production, and triggering apoptosis or necrosis. Alterations in amino acid and lipid metabolism contribute to steatosis or fat accumulation in the liver. Copper also disrupts lysosomal exocytosis, the process by which lysosomes expel cellular waste. This disruption prevents hepatocytes from eliminating excess copper, compounding toxicity, according to Poschluck, published in 2014. These cellular changes trigger inflammation and recruitment of immune cells, which contribute to the development of fibrosis and cirrhosis over time. Cellular damage begins long before symptoms appear, highlighting the importance of early detection and intervention. Hepatic manifestations. The liver is the first organ affected in Wilson's disease because it is central to copper metabolism. Early disease may present as hepatomegaly, enlarged liver, or mild elevations in liver enzymes, but over time, copper-induced damages lead to chronic hepatitis, steatosis, and eventually fibrosis and cirrhosis, according to Husser. In severe cases, patients may develop acute liver failure, sometimes accompanied by Coups negative hemolytic anemia caused by copper-damaging red blood cell membranes, according to Allah, published in 2007. Long-standing diseases increase the risk of hepatic cellular carcinoma. Liver involvement often occurs before neurological symptoms develop, making hepatic signs a key early indicator of Wilson's disease and an important diagnostic clue. Neurological and ocular manifestations. When copper storage capacity in the liver is exceeded, copper enters the bloodstream and crosses the blood-brain barrier, where it accumulates in the basal ganglia, particularly the putamen and globulus pallidus. Damage to neurons in these areas leads to movement disorders, including tremors, dystonia, and slurred speech. Patients may also experience Parkinsonian features due to basal ganglia dysfunction. In addition to motor symptoms, many patients develop psychiatric symptoms such as depression, mood swings, cognitive decline, and personality changes. Copper also deposits in the cornea, forming Kaiser-Fleischer rings, which appear as golden brown rings around the cornea and are present in over 90% of patients with neurological involvement. These ocular signs are highly specific and can provide an important diagnostic tool. Systemic impact. Wilson's disease affects multiple organ systems beyond the liver and brain. In the kidneys, copper accumulation can cause Fanconi syndrome, leading to the loss of amino acids, phosphates, and glucose in the urine. The heart may develop arrhythmias or cardiomyopathy due to oxidative stress and structural changes in heart muscle. Copper accumulation in bones and joints can result in arthritis and reduced bone density, according to Badman, published in 2015. The endocrine system may also be affected, causing menstrual irregularities or infertility. Psychiatric symptoms such as mood disorders or cognitive decline may appear even before neurological signs, emphasizing the importance of early detection. This widespread organ involvement reflects the copper's essential role in many biological processes and the broad consequences of its dysregulation. Diagnosis. Wilson's disease can be challenging to diagnose due to variable presentations and overlap with other liver and neurological disorders. A combination, of biochemical tests, clinical findings, and genetic testing is usually required, according to Shishlik. Serum seroplasm is typically low because copper is not incorporated into the protein. 24-hour copper excretion is elevated as the body attempts to excrete excess copper. Slit lamp examination can detect Kaiser-Fleischer rings, which strongly indicate Wilson's disease, especially when neurological symptoms are present. Liver biopsy measures copper concentration directly, and values above 250 dry rate when confirmed with significant copper overload. Genetic testing for ATP7B mutations is a definitive diagnostic tool, especially when biochemical tests are inconclusive or for family screening. Early diagnosis is critical because starting treatment before organ damage occurs greatly improves outcome. All right, so the treatment. The main goal is to remove excess copper and prevent further accumulation according to Cichlick. Chelation therapy is a primary treatment and involves medications that bind copper, allowing it to be excreted in the urine. D-penicillamine is one of the most commonly used chelators but can cause side effects such as allergic reactions and kidney problems. Trietine tetrahydrochloride is an alternative with fewer side effects and has been shown to equally be as effective according to Cichlick. Zinc therapy is another option that works by inducing metallothionein, a protein that binds copper in interstitial cells, preventing absorption, according to Boll. Zinc is often used for maintenance therapy or in asymptomatic patients. In cases of acute liver failure or advanced cirrhosis, liver transplantation is necessary and corrects the underlying metabolic defects because the new liver contains functional ATP7B. Treatment must continue for life as stopping therapy leads to rapid copper buildup. Regular monitoring of copper levels, ceruloplasmin, and liver function ensures therapy is working effectively. The roles of allied health professionals. Managing Wilson disease requires a multidisciplinary team. Dietitians play a key role in guiding patients through a low copper diet, advising them to avoid copper-rich foods such as shellfish, organ meats, nuts, chocolate, and mushrooms, according to Husser. Physical therapists help patients improve movement and coordination affected by neurological symptoms. Occupational therapists assist with daily activities, and speech therapists address speech problems caused by neurological damage. Dialysis technologists may be involved in plasmapheresis, a proved seizure that removes copper from the blood during acute crises. Mental health professionals support patients and families in coping with the psychological impact of living with a chronic condition. This collaborative approach improves the quality of life and enhances treatment outcomes. Trends in research. Current research is focused on developing new treatments and improving early detection. Gene therapy is a major area of exploration and aims to deliver a functional ATP7B gene to liver cells. Animal studies using adeno-associated viral vectors have shown that gene therapy can restore copper balance and reverse liver damage, according to Cichlick. New chelating agents with fewer side effects and better ability to cross the blood-brain barrier are being developed to improve treatment of neurological symptoms. Advances in genetic testing are enabling earlier and more accurate detection of ATP7B mutations for research on newborn screening, such as the previously mentioned dried blood spot seroplasm assays, may allow diagnosis before symptoms appear, according to Mack, published in 2022. These efforts could transform Wilson's disease from a lifelong condition to one that is even treatable or even curable. In conclusion, Wilson's disease is a genetic disorder caused by mutations in the ATP7B gene, which disrupt copper transport and lead to toxic copper accumulation. This accumulation causes oxidative stress, mitochondrial damage, and cell death, resulting in liver disease, neurological problems, and other complications. Early diagnostics through biochemical testing and genetic analysis is essential, and lifelong treatments such as chelating agents, zinc therapy, and liver transplantation can control copper levels and prevent serious outcomes. Ongoing research into gene therapy, improved chelators, and newborn screening offers hope for more effective treatments and potential cures. Wilson's disease illustrates how a single defect in cellular transport can disrupt homeostasis across multiple organ systems and how advances in molecular medicine are helping to manage and potentially eliminate this condition. Thank you so much for coming to my presentation. These are my references. If you have any further questions, you can email or reach out to me with those questions, and I'll answer them as soon as possible. Thank you so much, and have a good day.